Okay, so why don't we get started? Today's lecture is going to be focused on the auditory system, the wonderful world of sound. But before I do that, what I wanted to try and do is emphasize some of the key similarities and differences in the two major sensory systems we've gone over so far. I think Professor Hawken did a fantastic job on the visual system. One thing he didn't say explicitly, but you should all be aware of that from the volume of information that he conveyed to you, is that the visual system is the system that we know the most about. It's the system that we studied for the longest amount of time. And so for that reason, that's why those lectures were particularly dense and going into more detail on every single pathway between sensory transduction all the way up to topographic maps and beyond to things like prosopagnosia. Okay? But I wanted to try and emphasize some both similarities and differences because the reason why I've emphasized these key principles is to help you learn these systems better so that you can have kind of a framework for understanding how these different sensory systems work. And so let me just jump back first to the somatosensory systems where we talked about transduction, detection of outside energy, just the force of energy that we feel through all these different receptors that we have in different densities across our bodies. And of course, we talked about the hands and the lips being areas where there's a high density of these types of receptors. And going into transduction, we had a pretty simple and quite elegant way to transduce physical pressure into the mechanically gated ion channel, where you press down on it, and the channels actually physically open upon physical movement, and they let sodium channels in, and you get a nice depolarization. First followed by generator potential, something like an EPSP, and then once it reaches threshold, you get a regular action potential out of the sensory neurons. Okay, now let's contrast that with what Professor Hawken told you about the visual system. Here we have visual light energy coming into the eye and going into the retina, traveling through the layers to the back of the eye where the rods and cones are. And we talked about really elegant set of chemical and molecular the rods that allow transduction to take place. And I want to just emphasize a major difference between the somatosensory system and the visual system that Professor Hawken talked about in the visual system, but he didn't make the comparison. Actually, he did partially, but I just want to emphasize it. And the comparison is this. The mechanically gated ion channels depolarize when they are stimulated. You touch the fingertips and you get a depolarization through pathways that you've studied and you're very familiar with. Sodium ions coming through the channel causing a depolarization, an increase in the potential from negative to positive, okay? Something very different happened in the visual system, right? So light is coming onto those rods and cones, and what does it cause? Not a depolarization, but a hyperpolarization, okay? That's very, very different. So where does that signal switch? Does it switch to a depolarization? And if so, where does it switch? Anybody? Does everything stay depolarized throughout all the different pathways that he talked about? Or is there other cells that depolarize instead of hyperpolarize? This is the major signal coming through those rods. What do you think? Yes? Right. Exactly. So basically, this signal gets switched to depolarization at the next synapse from the rods or cones to the bipolar cells, okay? But the main difference I want to emphasize here, light energy is causing hyperpolarization in the visual system, whereas it causes depolarization in the somatosensory system. A big difference, and it's reversed at the next synapse for the light, but this is a huge kind of shift in the way that these neurons are responding in the visual system compared to the somatosensory system. Okay, so there are a couple of things about the visual system that are a little bit backwards. Can anybody think of another backwards kind of response or pathway that Professor Hawken talked about in the visual system that is just opposite from what you might expect in other sensory systems, something a little bit opposite? Any ideas? What if I show you this? Anything backwards or a little bit odd about the retina? Where is light coming? From here or here? Here? Yes. Right, exactly. The receptors are at the back of the retina. Why would you organize a system like that where the light that you're trying to see has to go through all these cell layers before it gets to the receptors that are sensing it? Okay, there's no blockage to mechanical receptors that are on your body. Okay, it's directly, the physical energy is going directly to those receptors. But in the visual system, it's evolved to have this unusual kind of organization. It seems to work just fine, but still just backwards from an engineering point of view. Does everybody get what I'm saying? That is odd because it's just backwards. So there's a lot of unique things about the visual system. The hyperpolarization, light causing hyperpolarization in the cones and rods, and this backwardness of how light has to travel through all these cell layers before it finally gets to those very sensitive rods and cones at the whole back of the eye. Okay. I'm just trying to I'm not going to go over every single point, but one of the questions on your list of questions for this lecture is to list on your own, not the examples that we went over, but other examples that you think about in terms of similarities or differences between the auditory and the somatosensory system. Okay. Receptive fields, very, very important. And here, I would strongly recommend you, again, look at those animations because those were a beautiful illustration of the center surround organization of the receptive fields. So I think you can understand, and the first example we started out with was one that I think is quite easy to understand. So if you had an animal, let's say you were trying to record my somatosensory receptive fields, and you had a way to non-invasively record from my primary somatosensory cortex. What would you do to actually map out the receptive field of whatever cell you happen to record from? What would you do? Any idea? Yes? How would you know where the receptor was? And everybody can feel free to help in the answer of this question. Yes? That's a good point. So she said, can't you kind of determine where it is since you know where you're recording? But what if you don't know? There's not little black mark showing you your little homunculus up there. It's just a brain, and you happen to stick an electrode in. That's what you do as a neuroscientist. So you stick it in. You don't know where you are in this homunculus. Maybe it's an animal you haven't recorded from before. Okay, so it's me, but you haven't recorded from me before. So what would you do as a scientist? You're trying to map out the receptive field. Yes? Exactly. One of the options is just to feel everywhere. You don't know where it is, so you're going to have to touch all over the place to see what area of my body, when you touch it, is going to react with a response in the neuron that I happen to be recording from. Okay, so you are now a bona fide electrophysiologist. You know how to do this experiment, but this is for everybody. You need to be able to understand this. I want you to understand not just this picture, but what you would have to do to actually get this response, okay? So I am recording in this cat's cortex, and I don't know, there's no little, bullseye on the cat telling me where his receptive field is. You have to go and figure it out. It could be here, it could be on the tail, it could be on his neck. You have to feel it out, and then what you have to do is map by pressing with little points and bigger points the whole extent of this cell's receptive field, okay? So that is how you map a somatosensory receptive field. Yes, question? Okay, that's all right. Any other questions? Does everybody get that, okay? So a question that I might ask you on the exam is exactly that. You are a physiologist. Describe for me how you would map out a receptive field that you think may be on the right arm or the left arm, okay? Something like that. You have to understand where you're putting the electrode and what you're testing in order to map a receptive field. Does everybody get that? Is that clear for everybody? Okay, if it's not, either come and ask me later or go talk to your TAs, because this is a very, very important concept. And I want to follow this up by the visual receptive field. Okay, so I'm going to ask you exactly the same question. Now I'm recording from area V1. Where is area V1 in the brain? It's primary visual cortex, but where is it exactly? You said that. Front of the brain, back of the brain, side of the brain, inside the brain. Where is primary visual cortex? Back of the brain, exactly. What lobe is that called? Occipital lobe. Okay, so you're recording from an electrode in the occipital lobe. You happen to be in primary visual cortex. Okay, let's say you're doing it, again, on me. How exactly, what exactly does this mean? How are you mapping out the visual receptive field? What are you doing with me to actually map this out, do you think? What is the exact setup? How are you stimulating the visual receptive field? Okay, we just talked about how you stimulate the somatosensory receptive field. What about the visual receptive field? How do you do that exactly? Yes? With light. Where? So are you shining a light in the eye? Maybe. Okay. Anybody else have an idea? Do you shine a light in the eye in a circular form this way? What's stimulating? Can you all see me? Okay, good, excellent. Yes, you can all see me. Am I shining a light in your eye? It's a stimulation out here. You are seeing my form because of the differences in light, because of the shadows, okay? So what I would do is not shine a light in your eye, but I would shine a light here to stimulate what is exactly going into your visual system right now. So actually, there's an animal. How this actually works, what this means is there's an animal with its eyes fixated on a certain point. The eyes are not moving, and what they're doing is they're shining a light on the screen, just like in a dark room, okay? So the animal's in a dark room, and what you do is make sure, let's say it's a human animal, you ask the animal to, your subject to fixate on a central spot, right here where my red pointer is, and then you're stimulating this person's visual receptive field. You guys are my visual receptive field, okay? So if I wanted to stimulate, I wanted to test a visual receptive field, I would fixate here, and let's say you, right there, that I'm pointing my pointer on, you would light up, your head would light up, okay? So I am fixating here, and your head is lighting up, and you are stimulating one part of my retina, okay? So it's not the light, I'm not shining a laser light in here, I'm letting that light out there come in and stimulate my visual receptive field, okay? And I do that with small points of light. So it's the visual receptive field out there. This is your visual receptive field, okay? This is my visual receptive field. I am your visual receptive field, just like doing this is stimulating my somatosensory receptor, okay? So does that make sense? So that's exactly how you would, as a physiologist, start to test this. You would make an animal fixate, you record a neuron in primary visual cortex, and then you could map out in visual degrees of angle exactly where you're stimulating with small little points of light. And that way, you can stimulate here, and you happen to get the center excitatory part of a visual receptive field, and you get this response. And then you go a little bit further, and then it's actually inhibited. And then you map out exactly this center surround activity, but you're stimulating out in the animal's own visual receptive field. Okay? Is that clear? Okay? So you should be able now to describe for me in a short answer paragraph format how you as a physiologist would map out visual and somatosensory receptive fields, and also tell me the similarities and differences in that process between the between the visual and the somatosensory system. Again, look at what the animations don't show you is the exact setup that you're using. So that's what I wanted to make sure that you understand. And then lastly, another nice parallel that we talked about is in the level of the topographic maps. So here is your very familiar somatosensory homunculus with large parts of the cortex representing areas where the most somatosensory information is. That is the hands, and also the lips are highly represented. So we have a overrepresentation of certain parts of the body. Now let me ask you, is there an overrepresentation of certain parts of the visual environment? Yes? In the center, in the fovea, exactly. So that's exactly parallel. The most sensitive parts of our somatosensory systems are our hands and our lips. The most sensitive part of the visual cortex is the fovea. That is the center of my attention. I'm moving my fovea around so I can fixate on you, and I can see your facial expressions, and I can tell whether you're interested or paying attention. But I could tell people are over here, but I really can't see what their visual expressions are. Why? Because the fovea has the most sensitivity in terms of identifying details. And that is represented in, you can't see it here, this is the location of primary visual cortex, but Professor Hawkins showed you a nice figure that you can go back and look at how much more of the cortex is devoted to processing the foveal representation compared to the extrafoveal representation. Okay, and then finally, I just wanted to try and bring it back to something that you're very familiar with. We talked about higher order visual areas. And not only is the visual system the system that we know the most about, but it's also the system where so much of our cortex as Homo sapiens are devoted to processing. A huge proportion of our cortical areas process vision or higher order vision. And one of the areas that we talked about is the fusiform gyrus down here on the ventral surface. Anybody remember what this fusiform gyrus was important for processing? Faces, exactly. And so he showed this fMRI study where you get a higher activation for visible faces here than to visible houses, and then another area, the parahippocampal place area, that is selectively active to houses and not responsive to faces. So a specific area is important for this thing that's so ethologically significant, being able to recognize different faces. And this I will just bring back to what we talked about in the very first lecture, prosopagnosia in the artist Chuck Close, who is able to paint in these amazing ways and put this 3D face into a 2D environment. And I don't think I've never read anything that Chuck Close has ever been scanned in an MRI to see what the organization of his fusiform gyrus is. But presumably there are impairments or differences in this area of cortex in his brain that does not allow him, even to this day, to recognize individual faces. Again, he says that I would highly recommend, I don't remember whether I told you this, but there's a great interview that Chuck Close did with Stephen Colbert that's both funny but also very informative about how he sees faces. And his comment was, if you stay still, I could get a feeling for what you look at. But the moment that you move and I see a different aspect of your face, you look unrecognizable again. So that's everything and more that this fusiform gyrus is doing. So I think at this midpoint of the class, hopefully you can see how much you now know about some of the things we talked about in the very first lecture. Areas important for visual processing, but also specifically important for face recognition, you have a much deeper understanding of now, presumably than you did at the beginning of class. Okay, so let's jump to the auditory system. So the visual system is amazing for our ability to be able to perceive and recognize and separate so many different visual things in the environment. But the auditory system is equally impressive. And we're going to be talking about, towards the end, about people that enhance this auditory system, people like musicians, people like conductors. But I just want to do a quick test with you. Just to illustrate the power of your own auditory system. So this is really asking about your higher order auditory perceptual qualities or abilities. And if you read the very first introductory part of this chapter on auditory system, you'll have read a little part that says, you know, there are certain songs that people can recognize in only a single note. Okay, so this represents, you know, the power of the auditory memory and the auditory system. So I'm just going to test you on these different things. So I'm just going to give you a note and see how many people can recognize these different songs. Okay, we're going to start with this one. Ready? Hard Day's Night, excellent, one note. Next one. Yes? Very good, okay. Next. That was easy. Last one. Without You, very good. Okay, you all passed. Amazing. So that was literally the first couple of notes of several songs, both older songs as well as newer songs. And all of that auditory information is obviously seared into your brain so that you can recognize it after only one note. So that's what we're trying to appreciate here. How does our auditory system allow us to do that? Of course, we're going to start with the simple stuff. How sound get into the system, but even earlier than that, what is sound? Okay, so sound is really made up of vibrations of air, compressions and decompressions of air in a very regular way. And there are particular levels of vibration that are called pure tones. Pure tones are those tones that you measure with those little tuning forks, okay? And they have two major characteristics. They have an amplitude or intensity that's perceived as loudness. Everybody can appreciate loud tones versus low tones. And a frequency. Frequency refers to the number of cycles per second of those vibrations of air particles. And that's measured in hertz, cycles per second. And that frequency is perceived as a pitch. So high pitch is very, very high, and low pitch is low. And that really just has to do with the number of cycles per second that the air is vibrating at. Okay? So here is an illustration of one of those tuning forks. We see here a visual representation of the compression and expansion of these little blue dots, which are the air particles. And here is a graphical representation of those regular vibrations shown as sine waves. And you can see the amplitude is measured here, higher amplitude here compared to there, and the frequency is the number of these cycles that you get in a second. This is lower frequency, and here is higher frequency or higher pitch. Okay, so sounds contain fundamental or basic frequencies, and harmonics are multiples of that frequency. So any of you that are musicians may be familiar with this, particularly if you play string instruments. Timbre is characteristic sound quality of an instrument and relates to the intensities of the different harmonics. So let me just go very, very briefly through harmonics. Here is a fundamental frequency, and then you can see that the different harmonics are exactly a third of the amplitude of this, a fifth of the amplitude, and the sum of all three. And we use something called Fourier analysis. It's a mathematical process that's used to analyze sounds as the sum of the sine waves. So you can sum up the fundamental frequencies together with the harmonics mathematically through Fourier analysis. And of course, all the sounds that we are used to listening to, the sound of different people's voices, and how you could recognize the voice of your mother or your father with just a small snippet of that stimulation. Or all the music that we're able to recognize in only a single note. Very, very complex multiples, so frequency as well as harmonics. Okay, oh, and the last point, the decibel is a measure of sound intensity. You're probably familiar with that. Okay, so how do you get sound that's compression and decompression of air into the auditory system, encoded in a way that we can make sense out of it? Okay, so we start with the external ear, the pinna it's called. These are the pinna are our earlobes, and there are lots of different types of pinnas, really big ones, smaller ones, bigger ones that way, ones that point out and move around, and then we're going to talk about the back a lot in the last section. What is the goal of the pinna? The pinna is really funneling sound into your auditory system. It works to help to bring the sound and concentrate it at a frequency that is most sensitive for your auditory system. So the bat will take in sound at a very different level, because it's actually not only hearing external sounds, but it's hearing echoes off of external things in the environment that it uses for something called echolocation that we're going to be talking about at the end of the lecture today, or possibly on Wednesday. Okay, so we have a funneling of sound through the external ear, particularly through the pinna. So the external ear, or pinna, collects the sound waves, and the shape of the external ear transforms sound energies. Our ears are shaped the way they're shaped because they're helping us transform the sound information into frequencies that we are most sensitive to. And we're most sensitive to those frequencies that are associated with speech. Okay? So here is the external ear. The external ear is made up of both the external ear, or pinna, as well as the ear canal. The second major part of the ear, external ear, second major part is the middle ear, critical part of the ear, and it's made up of the tympanic membrane here, or eardrum right at the end of the ear canal. That's why you're not supposed to clean out your ears with those long, puffy things, because damaging this, as you'll see in a moment, is really bad. As well as 3 teeny tiny little ossicles or bones, okay? So let's look at the detail, the blow up of this middle ear. And the purpose of the middle ear is to concentrate the sound. So the external ear kind of funnels sound, and then the middle ear is going to concentrate that sound through these little ossicles. Okay. The 3 ossicles have names that you need to know. First is the middle one is the incus, and the third one in the sequence, stapes. It looks like a stirrup. Stapes means stirrup. And these ossicles connect the tympanic membrane, the eardrum, to what's called window, part of the internal, the inner ear. Okay, it connects the oval window connects the middle ear to the internal ear. Okay, so here is a blow up of the middle ear. So we're blowing up just this part right here, the middle ear. Looks like this. Here's a big blow up of the tympanic membrane, the eardrum, right at the end of your ear canal. This tympanic membrane is physically connected to these 3 little ossicles. The malleus is here, right, connecting directly to the tympanic membrane. The malleus connects to the incus right here, and the incus connects to the stapes that is pushed up against what's called the oval window. Okay? So what happens here? What you have is a translation of the vibrations of the air that are vibrating the tympanic membrane, and then they're concentrated because those vibrations get translated into little vibrations of all these 3 little bones that then translated into vibrations onto the oval window. Okay? So you have sound information translated into physical vibrations of these bones hitting the oval window. Okay? That is how the middle ear concentrates sound. Now, 2 important muscles that I want you to know and understand. These are 2 muscles. One of them is here, the tensor tympani muscle, and it's connected to the malleus. And the other muscle, smaller muscle, is called the stapesius muscle. What happens when those muscles are contracted is that it stops the vibration of these bones. When does it happen? It happens when you sneeze. It happens when you cough. It happens when you talk. Why? Because if you didn't decrease the sound level within your ear, if you didn't stop these bones from vibrating when you sneezed, it would sound like an explosion in your head every single time. Okay? And it also helps if the music is too loud. I guarantee if you go to a concert today, these muscles are tensed up to try and decrease that huge amount of sound that's typically at concerts. It's a safety mechanism. And it's particularly valuable for, our voices can be pretty high. I'm sure these muscles work a lot in opera singers that learn how to sing very, very loud. But muscles are particularly important for animals like bats. Bats emit very, very, very strong calls for the echolocation that they do. It's at a higher frequency that we can hear, so we can't really hear all their frequencies. But every time they make those calls, these muscles within their ear canals are tensing to actually decrease that sound volume. Okay, here, so you can see the spelling of these, the tensor tympani and the stapesius, and they stiffen to reduce sound effects. Okay, so now we're at the inner ear. So now we have the vibrations in the air translated into vibrations of these little muscles, and it is hitting the oval window. The oval window is connected to the inner ear, which is a fluid-filled space. And so what this little space does is it hits it's like hitting a waterbed. When you hit a waterbed, what happens? You see the ripples, right? And so the fluid-filled chambers don't have any place to go. And so when this starts hitting here, there is an outlet for that vibration that it's hitting the next section with, and that is this window right here called the round window. So every time this hits here, the round window pops out like that because there's a counterforce for every force of vibration that the stapes is hitting the oval window, because this is a fluid-filled space inside this inner ear that we're going to look at in a second. Okay, where is the inner ear? This is the inner ear, and before I show you the structure, which is very complicated, and here, yes? Does fluid come out of the round window? No, that's a good point. Fluid does not come out of the round window. This is covered. It's just the force. So imagine a waterbed, and it just goes up and down. So what you see is this popping out and in, okay? Fluid doesn't come out. You don't have, you know, like wet ears every time you hear something. Okay, so let's talk about the inner ear. The inner ear converts sound to neural activity. So here you're familiar with this. All of these sensory systems have a particular structure that is helping you convert external information into neural activity. And in the auditory system, it's the inner ear. Mammals, the main structure within the inner ear is called the cochlea. It's a spiral structure with a base and an apex. And the base is nearest the oval window, and it goes up in like a little shell form up to its apex. Okay, the cochlea has three main parts. You need to understand and know the names of these three parallel canals within the cochlea. They're called the scala vestibuli, vestibular canal, the scala media, the middle canal, and the scala tympani, the tympanic canal. We're going to focus, you need to know all three names, but we're going to focus on the scala media because that's exactly where all the action is taking place in the auditory system. We already talked about the round window. It's a membrane that separates the scala tympani from the middle ear, and that's where the extra wave is coming out. Okay, so here is these three little chambers that are going around this kind of rolled up circle. This is the scala vestibuli, the scala media, where all the action takes place, and we're going to look at this in higher magnification, and the scala tympani, the tympanic canal. All three fluid-filled, but again, the action is really taking place right here in the scala media. So let's take a look at exactly what that looks like. The scala media is home to a key organ in the auditory system called the organ of Corti. The organ of Corti converts sound into neural activity and has, again, three main structures: sensory cells or hair cells, framework or support, and the basal membrane. And the basal membrane is what's vibrating in response to the sound. It's vibrating in response to this stapes hitting the oval window. Okay? So let's look at these three main parts of the organ of Corti within the scala media. Here's what it looks like. Here's the scala media. Here is the scala tympani. And here is the organ, major components of the organ of Corti. I said the organ of Corti is made up of hair cells. Here are hair cells. There are two flavors: inner hair cells and outer hair cells. Here are supporting cells over here. And here, down at the base, is the basilar membrane. There's another key component that I didn't write down here, but it's in your book, and that is the afferent fibers, those fibers that are bringing the neuronal information from the hair cells into the central nervous system. That's also a critical part of the organ of Corti. The other main component of the organ of Corti, that's not a part of the organ of Corti, but critical within the scala media, is another membrane called the tectorial membrane. You need to know the difference between the basilar membrane, part of the organ of Corti, and the tectorial membrane, not part of the organ of Corti. So let's talk about what those two different membranes do. Sound vibrations cause the basilar membrane, part of the organ of Corti, to oscillate. And the cool thing about the basilar membrane is that different parts of the membrane are sensitive and respond to different frequencies. So high frequencies displace a part of the basilar membrane right near the base of that cochlea. And low frequencies displace a wider apex. So here is the cochlea unraveled. Here's the base and here's the tip. The tip is curled up here. And the basilar membrane, if you were to splay it out, actually goes for several millimeters. And this is called the base, right next to the stapes here, right next to the inner ear. And this is the apex. So what they found is that different parts of this basilar membrane will move, will oscillate, depending on what frequency is being pounded out here at the oval window. If it's a high frequency, you get oscillations and movement in this part of the basilar membrane. However, with very low frequency stimulation, 25 hertz, you get activation and movement here at the apex of the basilar membrane. Okay? So you have very high frequency stimulating here, and low frequency doesn't move anything until you get out to the very apex of this basilar membrane. Okay, so that is what's moving the basilar membrane, but we still haven't got to the actual transduction. As I mentioned, the organ of Corti has two sets of sensory cells, the inner hair cells, IHCs, and the outer hair cells. Each hair cell, both inner and outer, have what's called stereocilia, or hairs protruding from each cell. A critical part of these inner and outer hair cells are the afferent and efferent nerve fibers that carry messages between the hair cells and the brain. Different neurotransmitters are active, and I'm going to tell you exactly what I want you to know about this afferent and efferent information. The critical part is written down here. Of these two hair cells, if you were to have to lose one of them, no question, you want to lose your outer hair cells. Why? Because the inner hair cells that account for 95%, this should be 95%. I'll change that on the outline. 90 to 95% of afferent auditory fibers and give rise to the perception of sound. So it's only the inner hair cells that are giving you all that stimulation through the primary auditory nerve that we'll talk about in a minute. The outer hair cells are not contributing to sound, but they're still useful. While they account for only 5 to 10% of the afferent auditory fibers, they're important for modulating sound. So while you can still hear, if you only have inner hair cells and not outer hair cells, your fine detail of hearing, if you were a conductor, to be able to tell the difference between what the first violinist and the second violinist is playing in a symphony, you would need absolutely your outer hair cells in addition to your inner hair cells. So let's talk about why that is the case. Here is another bigger picture. Here's the basilar membrane. We're in the scala media. These are the components of the organ of Corti. The basilar membrane, inner hair cells, outer hair cells. Here are the hairs, or stereocilia. And here, down here, are the afferent and efferent fibers. Afferent fibers are those fibers, and this is important for you to understand, afferent fibers are the fibers that are bringing information from the hair cells into the brain, into the cochlear nucleus of the brainstem through the auditory nerve. Efferent fibers are those fibers coming out from the brain, out to influence either the inner or the outer hair cells. And both the inner and outer hair cells have both afferent and efferent fibers. That's all I'm going to ask you to learn. You don't have to learn which neurotransmitters go in there. You do have to learn that there are many more many more outer hair cells than, no, that's not true. Many more inner hair cells than outer hair cells, okay? Now we want to look at the exact transmission or translation of this basilar membrane vibrating into action potentials. How exactly does that happen? And this is actually also very, very cool. It's like a feat of engineering, similar to the visual system. Auditory transduction, turning movement of the basilar membrane into action potentials. Okay, remember, only certain parts of the basal membrane are actually going to move in response to certain frequencies of sound. And the key here are these hair cells. Okay, these are the hairs. The hairs or the stereocilia on the hair cells. And what we're going to see is that sound displaces the hairs. How does it do that? These hairs are stuck up into this other key membrane, the tectorial membrane, okay? And so let's say this part of the basilar membrane is sensitive to high-frequency stimulation, high-frequency sound. And we have high-frequency sound coming in. This basal membrane starts vibrating because it's sensitive to that level of sound. Tectorial membrane stays still. That means that these cells, the hairs, are going to be displaced because this membrane is moving, this membrane is staying still, and my hairs are actually kind of stuck up into the tectorial membrane, as you can see depicted here. Okay? So what happens? My hairs move. The hairs move and something happens because here is a very, very, very high scanning electron microscope of 3 different hairs on a hair cell. And separating, or connecting, I should say, the 3 different hairs are these tiny little fibers called tip links. The shortest one is connected to the middle one. The middle one is connected to the highest one. So when all 3 of these hairs are displaced, what happens is the tip links pull. What do they do? Okay, sorry. Thin fibers called tip links run across each hair cell's stereocilia. Vibration makes the stereocilia sway, causes ion channels to open. So it turns out, that the displacement of the hairs causes pulling of the tip links, and the tip link pulling causes opening of channels that are indiscriminately sensitive to both calcium and potassium. Displacement of the hairs, because these tip links are literally opening these channels that are sensitive to both calcium and potassium that cause a depolarization in the hair cells. The depolarization goes down to the base of the hair cells where they stimulate voltage-activated calcium channels. So here's a theme that's going to come back again and again. Voltage-gated calcium channels now let in calcium down here that causes fusion of the vesicles in the hair cells and causes transmission. This is a depolarization going to the afferent fibers. Make up a lot of these were in the inner hair cells. These fibers down here are part of the auditory nerve. Okay, so that is exactly how vibration of sound in the air gets amplified in the middle ear, gets translated to a physical vibration within this fluid-filled inner ear. Only certain parts of the basilar membrane are sensitive to certain frequencies of vibration. The basilar membrane moves, and what happens? These stereocilia get displaced. And because of the tip links, you get a physical opening of channels and potassium. It's a little bit like the mechanical opening of the mechanoreceptors in your fingertips. This is happening in a very, very more elaborate way, but same kind of idea. And you come back to the same depolarization that you're all familiar with. Okay. Hair cells do. We talked about inner hair cells, and the inner hair cells are the ones that are really conveying most of the sound through the primary auditory nerve. What about the outer hair cells? Outer hair cells fine-tune the cochlea to permit finer discrimination of frequencies. And they can change the length of their stereocilia. And by doing that, that actually both stiffens the basilar membrane and it has effects on the tectorial membrane. So if you change the quality of both those membranes, you're going to change the sensitivity of your neighbor, the inner hair cell. And that can either fine-tune it or dampen it down. But in most of these cases, the outer hair cells are working to better tune the auditory information coming in. Okay. So I just want to emphasize the sensitivity. So, I mean, you look at this. These are very, very sensitive. You know, imagine all the evolution that went into the development of this very, very intricate system to allow us to hear something as detailed as human speech and be able to tell even if somebody has an accent or somebody has a speech impairment or somebody is a bit high talker or a low talker, still to be able to understand language, okay? And so we can visualize, here are actual human, healthy hair cells and stereocilia. And this is what you want your auditory system and your auditory hair cells to look like. You want those hair cells to be standing up, to be as sensitive as possible to all that sound coming in. But in fact, the auditory system is bombarded every day. All your little earbuds, all of the sound, all of that construction that's always on the street in New York, all those squeaky subway wheels that come down and you see people protecting their ears. And they're protected for a reason. If you have damage to your auditory system, your hair cells go from looking like this to looking like this. Isn't that sad? Just sad, right? And it's sad and scary at the same time. So if you measure the amplitude of conversation, it's about 60 decibels. Noise from heavy traffic is 85 decibels. Damage can occur, this kind of damage to your hair cells can occur from 120 to 150 decibels. That is the of like a Hell's Angel motorcycle that you hear that are so loud, or what do they call, Harley Davidsons? Firecrackers are at this level that can cause damage, and small guns, okay? But also, if you could hear your next door neighbor's music from his earphones, that is a little dangerous sign. You're getting close to 120 to where you actually can do long-term damage. These hair cells don't come back. Once they've gone down, they don't come back up. So very important to realize this and protect these hair cells because, as you've just learned, it's a really very delicate and very elegant kind of computation and physical transformation that they're doing for your auditory system. Yes? There is a lot of phagocytosis that can go on. I'm saying this in general. I don't know specifically whether they will eat up a dead hair cell, but in general, yes, there are lots of mechanisms in place to take away things that have died from your central nervous system. Yes? Yes. They just break. Yeah. Yes, I mean, too high of a intensity light into your eyes, that's why you're not supposed to sit there and stare at the sun for a long time. Yeah, that will damage your photoreceptors in a very similar way as this. But I would have to say that this might be the easiest sensory system to damage. And it's very, very difficult to to, you lose your ability to communicate when you lose your auditory system. And there have been studies showing that the increased use of the earbuds at higher volumes are causing much more auditory damage in young people today than they had seen in the past. So you really do want to be careful about that. Yes?
What's the threshold of these earbuds? Like how many times like an explosion happens, how many times does it take for these earbuds, for these cells to die?
Yeah, that's a good question. I don't have the answer to that. I mean,
how much loud music, how many concerts?
yeah, I don't know the exact answer to that. They just start getting damaged with exposure to this. Of course, the longer the exposure and the more intense, the worse the damage. But I don't know whether you can get a little damage and get a little bit damaged, but then you don't listen to loud music for a long time and it could come back. So I can find that out for you, but I don't know the answer. You and then you. Yes.
For people who are deaf, how does this play into it? Like, is it something that happens within the ear, or is it something within the mouth?
Yeah, that's a great question. So what happens when you're deaf? How does this play into it? And there are a number of different reasons for deafness. And there's actually a pretty nice chapter. I'm not going over it today, but you can read about the different reasons. And one reason could be damage or congenital malformation of the hair cells. But you can imagine that we talked about lots of different ways that you can damage your sound. If your ossicles don't work right, you're done. You can't get the right sound in. And actually, some of the most amazing research going on at the NYU Medical Center is in cochlear implants to actually reproduce the electrical activity of your living cochlea to be able to teach your brain to recognize even something as complex as speech. So if you're interested in that, I'd really recommend you go to the NYU Med Center website, to their cochlear implant section, and they have nice information about what they're doing, what you can do with cochlear implants. But it's using all of this neuroscience we know about transduction of auditory information to help deaf people, no matter how you became deaf. Yes?
If a gun goes off next to your head, something like that happens, there's like the thing where your hearing is impaired for like a couple of days, but it comes back.
Yeah.
So that's why it's not the hair cell thing that you other.
No, it does. It's still damaging the hair cells. So I mean, if you hit your head, you will have a concussion. You might see stars, and you could come back. You say, oh, I see pretty well, but you still probably killed a whole bunch of brain cells. In the same way that a gunshot really next to your ear will have definitely killed your brain cells, your hair cells. And you would have to go to an audiologist to just determine exactly what kind of damage has been done. I mean, it depends. Some people's hair cells could be more resilient than others, and it could depend on exactly how close it was. But the bottom line is just to appreciate the intricacy of what the hair cells are, you have to be able to appreciate, you do not want to damage these things because, they're not that sturdy.
What is ringing in the ear?
Yeah, ringing in the ear may be more of a nerve issue, a overstimulation of nerve, kind of like chronic pain that you feel, irritation of the nerves rather than a chronic stimulation of the hair cells. Any other questions? Yes, back there. Yeah. Yes. Yeah. That's a good question. I don't know the answer to that. I know that in everyday situations, in sneezing and in talking. So right now, my ear muscles are working, are stimulated. I don't know whether in a really traumatic situation like that, whether there's long-term contraction of those muscles and how that actually works. You can imagine that it might be, or everything gets messed up. You do a huge stimulation. I mean, what exactly happens when you have a concussion in the back of the head? There's just a lot of trauma. The same kind of trauma is happening when you have a gun go off right near your ear to your auditory system, and it's probably trying to react as much as possible, but I don't know if they know what the actual mechanism, long-term mechanism, or if there is a consistent long-term mechanism for those muscles after a traumatic auditory experience. Any other questions? Good questions. Yes?
When you're listening to yourself speak, like if you cover your ears and you can still hear yourself clearly, so
Yes.
does that activate like a different portion of the ear, or does it work the same?
Yeah, so she said if you cover your ears, you can still hear yourself talk. And it's activating the same portion because there's a lot of reverberation that can go on in your mouth. And so even though you close up your ears, there's probably still much more sound that gets in compared to sound from the outside. You're generating it all the way on the inside. There's actually a really cool picture. This one. Did you know that your cochlea was right behind this part of the eye? You just go back a little bit further. So it's all in there. I mean, it's strange to think about, but all this stuff is all packed in there into your brain, so or into your skull, I should say. So it's not surprising that the sound generated from your larynx can still activate all of these same things, even if you plug your ears. Yes?
Is there a difference between really loud high frequencies and really loud low frequencies?
Yes, there are differences in loud high and loud low frequencies. I think that, and it has to do with the sensitivity of the basilar membrane to intensities. I don't know all of the details, but there are differences there. And we're going to get to that in a second when we talk about sound localization, the differences between high and low frequencies. Any other questions? Okay, great. So why don't we go on? Okay, auditory pathways. So all of the afferent fibers from the inner hair cells that contribute to audition are going through the 8th cranial nerve, the auditory vestibular nerve. And we're not even going to go over the vestibular system, but just know that vestibular input comes through there as well. We talked about nerve 8. Vestibular cochlear nerve contains the auditory fibers from the cochlea. And each fiber divides into 2 branches, going to cells in the ventral and dorsal cochlear nucleus. All I want you to know is that the first stop of these auditory afferent fibers from nerve 8 is the cochlear nuclei. And this is, and I'll emphasize this, an interesting difference between the auditory system and the visual and the somatosensory system. So for the visual and somatosensory system, I emphasize the idea that the right part of our visual space is processed by the left side of our brain, and vice versa. My right hand somatosensory for my right hand is processed in my left somatosensory cortex, and there's this crossing over. We went over that. Where does it cross over? You need to know that for the visual and the somatosensory system. Auditory information is different. Lucky for you, you don't have to remember where it crosses over because it starts processing information from both ears from the very first relay point at the cochlear nuclei. Why? Because a large part of auditory information is localizing where that sound is coming from. And to be able to localize sound, you need input from both your right ear and your left ear. Okay? So what happens is, and you can see this here, this is a nice figure from your book. Here, this blue is auditory nerve 8, and it's synapsing in the cochlear nucleus. And you can see that information from the cochlear nucleus is not only from the, let's say this is the right ear. The right ear is not only receiving the right cochlear nucleus is not only receiving information from the right ear, but the right ear also projects directly to the left cochlear nucleus. So right and left are combining right at the first stage. So that's great. You don't have to remember where it crosses over anymore. It's all bilateral from the very beginning. What you do need to know is the first stage is the second stage, superior olivary, receiving it's called binaural information from both ears information. The next stage is the inferior colliculus. You remember seeing that from your sheep brains. It's a small little hump on the bottom two. There's two big humps, the superior colliculi, that are involved in visual processing, and the small two humps in the midbrain are called the inferior colliculi, processing auditory information. Here is that auditory information coming up. And then, like all the other sensory systems, this is similar to the other sensory systems, there is a relay through the thalamus. The thalamic nuclei, important for processing auditory information, is called the medial geniculate nucleus, MGN. The lateral geniculate nucleus processes what sensory system? What? Vision, yes, very good. So the lateral geniculate nucleus is for vision. The medial geniculate nucleus is for audition. So like all sensory systems, even though there's kind of mixing of right and left sides initially, all this information is processed through the thalamus, through the medial geniculate nucleus, and it goes into primary auditory cortex, which is situated on the lateral sulcus. Okay? So, cochlear nuclei have multiple targets, and so this is just saying in a slightly different way. There's superior olivary nucleus and the inferior colliculus, medial geniculate nucleus. And just like we had visual topographic organization, auditory neurons have tonotopic organization. What do you think tonotopic organization means? Any guesses? Yes. Organized by frequency, perfect. So you have low frequency processed right next to the next highest frequency, processed next to the next highest. So you go from low to high or high to low in a organized fashion. And that's exactly what this is, arranged in a map according to frequencies they respond to. And so here is an example of a slice through the inferior colliculus, one of the structures important for processing. And here is a visual representation of what this tonotopic organization in the inferior colliculus looks like. On this end, you have the low frequencies processed. So if I stuck an electrode in here, the cells in this strip would respond only to sounds in lower frequencies. And I would march across the inferior colliculus in this way and get higher and higher and higher frequencies. Tonotopic organization. Okay, so in the last 5 minutes, let's see if I could, okay, I'll try and get through this, but we're going to definitely come back. This is a really important concept that I want you to understand, and we're going to get cut off right in the middle, but I'll come back on Wednesday. Sound localization. Where is the sound coming from? Everybody can localize sound. Some people get really, really good at it, but everybody has the capacity to do that. And there are two major cues that you use to localize sound. One is intensity differences, okay? Differences in the loudness in the two ears. So if a sound is, somebody is talking over here, it will be more loud, that sound will be more loud in my right ear compared to my left ear. And my brain can compute that and say, oh, the sound is coming from over there. A second way the brain can compute sound localization is latency differences. So not only is a sound louder in this ear than this ear, but the sound reaches this ear sooner than it reaches that ear. So the auditory cells in this ear will be stimulated just a little bit earlier than that ear. And that is also processed by the brain, and that is used to localize sound. So for intensity differences. Here is a sound on the person's left side. The intensity is greater on the left-hand side, and the head itself, because it has a big shape, and those of you with large heads have even greater ability to have differentiation, because the head will cast what's called a sound shadow, blocking these waves of sound, and it'll make the perceived sound on the right ear just a little bit softer. So again, the intensity difference between the left ear, in this case louder, and the right ear, softer, will allow you to differentiate and say, hey, I know the sound is coming from the left. Okay, the second type of difference is latency differences, and there's actually two different flavors of latency differences. One is onset disparity, the difference in hearing the beginning of the sound, that's what I just described for you. But then there's an ongoing phase disparity that continues, which is defined as the continuous difference between the ears in the arrival in parts of the sound. So I think that's very clearly illustrated here. We'll end here. Here is the intensity difference, and here is a difference between low frequencies and high frequencies. High frequencies, when you're hearing high frequencies, there is much more sensitivity to intensity differences. There's almost no sensitivity to intensity differences in low frequencies. So those really low kind of bass sounds, it's really hard to tell where they're coming from. But a very high sound, if somebody has a really high squeaky voice over there, really easy to tell where that is, because you're much more sensitive about intensity differences at higher frequencies. Here are the two different types of latency differences. One is at the very beginning of the sound. It reaches one ear earlier than the other ear, and that difference can be seen. But you can see that the sound, once it gets there, processed by the two ears, continues to have an ongoing phase disparity. And that means that I could continue to tell this latency difference even after the very beginning of the sound. So both of these kinds of computations are used. And we're going to end there before we talk about how the brain uses this to tell where things are heard in space. Okay, see you on Wednesday.